By this time, you should have learned how all the cell parts work, what each of them does, and generally what they look like. Now we need to talk about how they actually work together to make things happen inside of a cell. So we're going to look at two different possibilities, and I want you to make sure you write them down, take notes as you watch this. If you fall behind, feel free to pause the video, back it up if you miss something, make sure you get all the notes written down. Okay, so two possibilities. The first possibility is we're going to look inside of a plant cell. Okay, we know that plant cells are different than animal cells because plant cells have chloroplasts that do photosynthesis. They capture sunlight energy, they use that energy to make ATP, and then to use that ATP to build sugar molecules. Okay, so that's the first cell part that's involved, is the chloroplast. And then once that sugar's made, it's got to get certain places. So if it's got to move throughout the cell, it's going to get transported by the ER, the endoplasmic reticulum, because that's the transport system for inside of cells. And it could go to one of two places. That sugar could get stored, okay, and it could get stored as starch molecules, and that would be stored in leucoplasts. They're like little baggies inside of cell that just store starch, okay, like a little vesicle that stores starch. Or those sugars could be used by the cell for cell respiration, and they could be burned to give the cell ATP, which is usable energy that it might need. Okay, so we see a total of 1, 2, 3, 4 cell parts all kind of working together here. That's just one possibility. Now we're going to move on to an animal cell. So if you remember the digestive system, the pancreas secretes digestive enzymes into the small intestine to digest food that's going through the small intestine. So let's say we're inside of a cell that's in the pancreas that makes those enzymes. Now these enzymes need to get secreted or squirted out of the cell, released from the cell into the small intestine. Okay, so they're not going to digest anything inside the cell itself. So we've got a whole series of different cell parts that are going to be involved. And the first thing is that in the nucleus of the cell, ribosomes get made. So they're made in the nucleus, and then they have to leave the nucleus through nuclear pores, little holes in the nuclear membrane, and they go out through that into the cytoplasm, where they then are able to attach to the rough endoplasmic reticulum. Okay, so remember that a cell is sort of like a water balloon with a whole bunch of stuff floating around inside of it. The water inside that water balloon will be like the cytoplasm, and that's where the liquid, where all the different interactions take place. So the ribosomes attach to rough ER. We know it's rough because it's got lots of little bumps all over it. Those are ribosomes. Okay, now these ribosomes are going to do what they do, and they make proteins. Okay, now those proteins get made and released directly into the ER, where they get sort of packaged up into a little storage vesicle and shipped out to the Golgi apparatus. Golgi is the major packaging and shipping center of the cell, so it's like the UPS of the cell. Once the proteins that are made, that are going to be our enzymes, they get to the Golgi, they get packaged up into a bigger vesicle, and then that vesicle can get transported out to get released to the outside of the cell. So they get transported along the cytoskeleton of the cell. There's two different types of cytoskeleton. There's microtubules and microfilaments. The microtubules would be the bigger ones, and this would be transported by a different protein and be carried along that cytoskeleton until it gets to the cell membrane. Okay? Once it gets to the cell membrane, the vesicle, which is made of a plasma membrane, which is very similar to the cell membrane, will fuse with it. It will open up, and whatever was inside that vesicle will now be on the outside of the cell, because the vesicle will become a part of the cell membrane. So technically the cell membrane will get just a little bit bigger at that moment. And anything that's inside gets released to the outside. This process is called exocytosis, and we'll talk more about that in the next unit. Okay? Now, we've talked about 1, 2, 3, 4, 5, 6, 7, 8 different cell parts here, all involved in just this process. And all of this is going to need ATP for all the energy to supply this, so the mitochondria is also going to be involved. So there's like 9 different cell parts here involved in making this happen to get that protein made by the cell and then sent outside of the cell. So now we're going to watch that video again, the Harvard University video, and I'm going to narrate what's going on here so we'll see the process that we just took notes on and how it's happening in real life. So what you're going to see at the start of this video is a blood vessel, and you'll see a bunch of red blood cells zipping by, and you'll see the white blood cells up at the top kind of rolling along the surface of, and they're looking for infections to try to fight, and they're going to attack and destroy whatever the infection is. Now we're zooming down inside of the white blood cell. We're looking at some of the components in the cell membrane and cellular communication going on between them and how this cell membrane is constructed. Chemical signals get sent through the membrane that would then tell the cell, hey, you found the right place to go and you can attack the infection. Now we're zooming down into the cell, and this is all microfilaments, all the little cytoskeleton that holds everything in place. And now this is microfilaments being built very quickly on one side of the cell, and on the other side of the cell they'd be cut and torn apart, and this is how the blob-like cell is able to actually move along. There's an enzyme cutting some of the microfilaments there. Here's microtubules being assembled very quickly on one side and disassembled on the opposite side of the cell as the blob moves forward along through the cell. Okay, here's a vesicle being transported by a transporter protein along that microtubule. And now they're going to show you kind of how that all happens. This is all cytoskeleton in the background. Here's a mitochondria supplying energy for all of this activity. This is the nuclear membrane, and here's the pores, and there are ribosomes being made that are coming out of the nucleus. They're attaching to messenger RNA, and the mRNA is going to get read by that ribosome, and a protein is produced. That protein might go bind with some sort of a carbohydrate or something to do something inside the cell. Again, ATP is provided by the mitochondria. Other ones will attach to the rough ER and produce those proteins directly into the ER, where they then would get transported throughout the cell where they needed to go until those proteins that were made would get to the Golgi apparatus. And here's the Golgi right here, where these are the vesicles, also called blebs, that will package up that protein like you see right here. Now it fuses with the cell membrane, it opens up, and everything that was inside the vesicle now becomes part of the outside of the cell. The cell then activates those proteins so they get folded to their correct shape, and they become communication proteins that stick out of the surface of the cell. When they communicate with a surrounding cell that tells them the signal that, hey, the virus or the bacteria that you need to fight is here, that's the signal for the white blood cell then to leave the blood vessel, go out into the surrounding tissue, gobble up and destroy whatever is causing that infection. All that happens within a split second inside of your cells, and that kind of thing happens millions of times per day. So that, in a sense, is showing you what we just went over here. It shows you all the steps that we just talked about. But everything doesn't always happen exactly the same in every cell. So the last thing to put in our notes is some stuff about cell specialization. So remember the difference. A generalized cell does generally everything that a cell needs within a single cell. So that's going to be things like, protists or bacteria or single-celled organisms, because they have only one cell. They have to be able to do everything. Otherwise, multicellular organisms like us all have specialized cells. And those cells have different types of organelles per cell, and even more different amounts of certain organelles depending on whatever the function of that cell is. Okay, so for instance, if you look in heart cells have lots of mitochondria. Why? Mitochondria, everybody could say, is the powerhouse of the cell. Well, what does that mean? It actually supplies ATP, which is the power or energy that a cell needs. Well, why does a heart cell need more energy than other cells? Because it beats about 1 trillion times in your lifetime. So there's constant muscular movement going on there and contraction of muscle cells. So you need a lot of ATP, therefore you need a lot of mitochondria. So heart cells have a lot more than other types of cells. Okay, lots and lots of lysosomes inside white blood cells. Well, why? What did we just talk about? White blood cells They find bacteria or viruses, they engulf them, they digest them, kind of like the blob. So inside of that cell, it needs to be able to break down and digest things. Well, that's what the lysosomes do. They would fuse with that bacteria and virus and then digest it inside the cell. If we look at liver and pancreas cells, there's tons of ribosomes in there. Why? Ribosomes make proteins. Okay, maybe they're making enzymes for digestion. Maybe they're making hormones like insulin, which is a protein. Okay, so because they make so many proteins, they tend to have a lot more ribosomes than other types of plant cells. Now, if we look at plant cells, for instance, most of you have eaten potatoes. Maybe you haven't seen an actual potato plant. The potato itself grows underground. There is no sunlight there, so there's no chloroplasts in a potato itself because it's actually part of the root of the plant. But if you let a potato plant sit out for a while, it will start to grow. Especially if you soak it in some water, it will grow leaves and a stem, and those leaves are the photosynthetic part of the plant. So they're going to have lots of chloroplasts in it. But once they make all that sugar, the sugar's then going to get basically stored. It's going to get linked together into starch and stored in the leucoplasts. So the potato itself is like a big starch storage locker for the plant. So it's got lots of leucoplasts in there to store lots of starch. That's why when you eat potatoes, you get a lot of starch. But the leaf cells themselves do not have a lot. They have a lot of chloroplasts instead, where there's none in the potato plant. So this is some idea of the specialization of cells, and they're going to be different depending on what their function is.